Up next is Byram Briddle. He's an associate professor of viral immunology at the University of Guelph in Canada. Historically, his research program had focused on a couple of areas, but particularly a novel vaccination strategy for cancers. And in response to the pandemic, Dr Bridle, in conjunction with some virology colleagues, has proposed to convert their cancer vaccine platforms into a novel vaccine technology against coronaviruses. And it's into that situation that we're invited in because New Zealand has an elimination strategy, which is apparently waiting for a vaccine. And we've had various dates that have been proposed for when such a vaccine would be around and available. And some of the developments in there that I think are particularly interesting for New Zealand, because if we are eliminating and if we are locking down, then we're waiting for a vaccine. And that's in which case that's a situation to which we invite Byram. Byram, are you there?
Yes, can you hear me, Mark?
Yes, we can. So fire away and we'll take questions for you at the end.
Sounds good. Okay, can you see my slide?
Can. Yes, we can.
Okay, great. And one final check, can you see the laser pointer moving around?
We can see that as well, yes indeed.
Okay, that's great, so I'll start. Okay, so thank you very much to the symposium organizers for inviting me to speak. I'm excited to be here. Welcome to all the participants. So I'm here to talk to you about COVID-19 vaccines and just want to give you an overview of all the facts, give an overview of what goes into vaccine development. And hopefully a lot of these facts will help those of you in New Zealand make informed decisions, especially those who are policymakers. So what I've done is I've set this presentation up so that I'm happy to provide a copy and will provide a copy to the symposium organizers. Anybody's welcome to request a copy from them, or you can request a copy from me. I've inserted into here some hyperlinks for some additional reading. I've got a table of contents here if you want to jump right to some of the key areas, and I'm going to jump right into it then. So first of all, in terms of who I am indeed an associate professor of viral immunology. I'm trained as an immunologist, have a PhD in immunology, and do a lot of work with viruses. And indeed, one of the things that we've been working on for years is using viruses as vaccine vectors to try and treat cancers. And so these vectors that we work with are very much sort of plug and play compatible. We can use these vaccines to target any protein that we want, including proteins that are derived from pathogens. So in other words, these vaccines can really easily be adapted to infectious diseases as well. And so indeed, that's what we've done. In fact, the type of immune response that is most effective against cancers is the same type of immune response that's most effective against viruses. And so in fact, my group has been commissioned by the government of Ontario to develop vaccines for COVID-19. So we have funding from that. We've also received funding from the institution that I'm at, and we're actively pursuing this. And I'm working, it's a large collaborative effort at my institution, and we've made very rapid progress. So in fact, we currently have 4 vaccines that we've developed, and we have several more that we're working on. And a couple of vaccines are looking very promising. And that's a term that you will have heard over and over and over again, and will continue to hear over and over and over again. And I can tell you very bluntly that even though we have some very promising looking vaccines, they are not going to help us with COVID-19 at all within the next 4 years at least. But you'll see why we're excited with this research when we get to the end of this presentation. We do think this will be valuable in the future. Okay, so one of the things I just want to go over first before I begin with the science behind vaccines. I just wanted to let you know that I teach the value of high quality and well-validated vaccines. The students that I teach can tell you I'm very passionate about it. I promote the use of vaccines. I really believe that vaccines are probably one of the best technologies that have ever come out of the field of immunology. It's the most efficient type of medicine. They can cost efficiently save millions of people from sickness and death. And so on one hand, some people wonder why, if I'm developing COVID vaccines, do I sort of speak out against all of the promises currently being made. It's because I really care about vaccines. Okay, and so I feel compelled to speak bluntly as an expert in the field. because for the sake of global health, we really need people around the world to maintain faith in vaccines, because vaccines are very important for maintaining global health. And if people lose faith in this through what I would argue is probably going to end up being, COVID-19 vaccine debacle, we don't want reemergence of diseases that are being well controlled now with current vaccines. All right. So in other words, I really worry about the potential damage to the credibility of the field of vaccinology if we keep pushing for unrealistic timelines. And I think you'll see in just a few slides what I'm talking about. So just to make sure we're on the same page, what we're talking about again is COVID-19, which is the disease identified at the end of 2019 called coronavirus disease. And it's caused by a virus called severe acute respiratory syndrome coronavirus 2. And this is numbered number 2 because the original SARS coronavirus outbreak that happened about 17 years ago, that was the original SARS coronavirus. Here's a picture of what the virus looks like. And what I want to do is, many of you probably didn't think you're going to be learning about immunology. Immunology can be very complex, but it doesn't have to be, your understanding of it does not have to be all that detailed in order to understand how vaccines work. So before we talk about vaccines, I just want to take you quickly through a couple slides. If you listen carefully to these few details about the immune system and how it works, then you'll really be able to understand what's important in terms of developing a good vaccine against SARS coronavirus 2, which again is the causative agent of COVID-19. So first of all, what you need to know is that the immune system is a complex collection of organs, cells, and molecules, and they all come together in the body to get rid of dangerous microbes, things that we would call pathogens. So these could be bacteria or they could be viruses. All right, and what you really need to know is that pathogens can be subdivided into 2 basic types. There's dangerous microbes that live outside of our cells, and that would encompass most bacteria. And there's another set of pathogens that live inside our cells, and those are viruses, like SARS coronavirus 2. Okay, so keep in mind that there's these 2 different types of pathogens, and that also means that an appropriate immune response against viruses is very different from the one that's most effective against extracellular bacteria, okay? So there's 2 types of immune responses that our body can initiate. And one of them is effective against viruses. The other one is designed to be effective against extracellular pathogens. So keep that in mind. Also, an effective antiviral response, so the one that we want against viruses, will induce 2 basic components of the immune system. T cells is one thing that we want, and antibodies. There's been a huge emphasis on antibodies during vaccine development, but there's no question that a really effective vaccine against viruses has to induce a balanced response. That means we want antibodies, and they have to be a certain type of antibodies, and we also need T cells, okay? So we need both of them. And this, again, is very important. So we're talking about the antibodies. For antiviral antibodies, the correct type is probably more important than the quantity. So a lot of people are focusing on how much of an antibody response they can generate with the vaccines that are being developed right now. But in fact, probably what we should be focusing on are what type of antibodies are being induced, okay? Because certain antibodies are better than others at neutralizing viruses, okay? So the second the last couple of things you need to know about the immune system is that the immune system needs to see 2 things before it'll produce these T cells and antibodies. So the first thing the immune system has to see is it has to know what the target is, all right? So in this context, it has to know that it's supposed to be targeting the SARS coronavirus 2. So the vaccine could contain the entire virus, or it can contain a piece of the virus, okay? But we have to show the immune system something from SARS coronavirus 2 so it knows what to target. And the second thing that the immune system always needs is it needs to know that that target is being seen in a dangerous context. So it needs an additional signal that's telling the immune system that, yes, this target you're seeing is from something that's very dangerous, and therefore it's worth responding to, okay? So keep that in mind. It's very important, because these 2 things that are needed for our immune system to respond to a virus we're going to come back to this in just a moment when we talk about vaccines. And finally, the other thing you need to know is the immune system has memory, and it literally functions like our the memory that we have embedded in our brains. So, but this is programmed into the immune system, and what this means is that once our immune system has responded to a pathogen, if it sees that pathogen a second time, it will respond much faster and much more robustly. And the consequence of the immune system responding much faster and to a much higher magnitude means that upon second exposure to a virus, our body can clear the virus before the onset of disease. So that's why for the vast majority of infections, once you're infected once, when you're infected the first time, you often get sick. But once you're infected a second time, you no longer get sick because the immune system has that memory and can respond so rapidly, it gets rid of it before we get sick. All right, so that's very important. That's why over time, as we develop immunity, we start becoming immune to more and more pathogens, and we don't experience sickness from them anymore. Okay, so that gives us a basic understanding of the immune system. So now we can focus on vaccines and then ask the question how these vaccines work. Okay, so first thing you'll notice right away, a COVID-19 vaccine must provide two signals. This should sound very familiar because this is exactly what we talked about. When the immune system naturally responds to a virus, it has to receive two signals. So what we want is a vaccine that can also provide those same two signals. So the vaccine has to contain the virus or a piece of the SARS coronavirus 2. And the second thing that it has to provide is that danger signal. So the immune system sees that SARS coronavirus 2 or a piece of it in a dangerous context. And that then causes the immune system to actively respond to it. Okay. And the reason why we're trying to develop a vaccine against COVID-19 is because a good vaccine will actually simulate the natural infection. And what that means is it'll cause your immune system to mount a response against that pathogen, but importantly, without causing the disease, because we're not actually infected with the causative agent. We're not infected with the live replicating SARS coronavirus 2. But the vaccine, a good vaccine, is designed to trick our immune system into thinking that our body is infected with this virus, so we respond to it. And now you can understand why memory becomes important, because the most important thing that a vaccine can do is confer this immunological memory. So we mount a response, and then our immune system remembers this pathogen. And then what happens is, the idea then is when a person actually becomes infected with the SARS coronavirus 2, right, when they actually get infected after vaccination, their body's seeing the virus, is infected with the virus for the very first time. But because of the previous vaccine, the immune system actually thinks it's seen the virus for the second time. And so this memory response is triggered, and the immune system will respond much faster and much more robustly than it did to the initial vaccination. And the idea is it'll respond so well, our immune system will respond so well that we don't get sick, that we clear the SARS coronavirus 2 and we don't get sick. And that's what would prevent ultimately spread of the disease from us to another individual. All right. And what this means is that vaccination is a way that we can accelerate progress towards what we call herd immunity. And this is the concept shown right here in this picture, where people in yellow are those who have been vaccinated, and so they are immune to the virus. And then we have individuals that are in blue, that are a few individuals, right? And these are people who were never infected with the virus, nor are they immune to the virus. Maybe these are people who chose not to take the vaccine, or these may be people who are vaccinated but did not respond particularly well to the vaccine, so they're not actually protected. And then we have people who were not protected, but got infected. These are red individuals. And so the concept with herd immunity is that once we have enough people who are immune, we don't need everybody to be immune, but there's enough people that are immune, those that then get infected with the virus and that could potentially transfer that to other people, chances are they're going to come into contact with people who are actually immune rather than people who are non-immune and therefore susceptible. And what this does, literally, the large group of people in the population that are now immune literally serve as a physical barrier between those who are infected and those who are uninfected but susceptible. That's how herd immunity works. What it means is we don't need everybody to be immune, but we do need most people to be immune. And for SARS coronavirus 2, the estimates have been anywhere from 60% to, most people would agree it's in the ballpark of 70%. Some argue that it's a dangerous enough virus that we need up to 85% of individuals to be immune in order to achieve this herd immunity and stop the spread of SARS coronavirus 2 through a population. Okay? Now, to get to the question that everybody's probably most interested in is, there's been promise after promise after promise, and this is why I'm speaking up, because I serve on so many grant review panels where people are proposing research programs to develop vaccine. And I can just tell you that the promises that are out there right now are generally incredibly unrealistic. So what I want to just take you through very quickly is so that you can understand what's involved in the development of a vaccine from the very beginning to when it would be distributed into people among the general public. So what we start with is exploratory research, okay? And this phase is focused on discovering, novel technologies to use as a vaccine and then developing these, and that can typically take 2 to 4 years. One of the things I want to point out, first of all, is that, of course, all these timelines that I have listed here are kind of typical average timelines, and they can vary dramatically. So just keep that in mind. But then what typically happens is once we have a vaccine technology that we think looks pretty promising, it has to go into the preclinical research phase where we start testing it in animal models. And ideally, we start optimizing the vaccine technology in these animal models. And that typically takes 1 to 2 years. Then there's a translational research pipeline. And this is where the vaccine is really looking promising. We have to start developing manufacturing tools, right? Ways to manufacture this such that they'll meet what we call good manufacturing practice guidelines, which basically means they're of a quality that could be used in people. And also there's going to be a lot of experiments that are going to use different animal models. And this is where often the use of things like non-human primates come into play to really focus on the safety of the vaccine and to try and generate additional evidence that the vaccine is going to work. And now this is really important because what I want to say is essentially at this stage it takes about 1 to 2 years. So what I'm going to say very bluntly is what I said about my own research. So the research that I'm doing right now is in the preclinical phase. I would argue that any vaccine that's currently in this first, second, or third phase of the research pipeline are not going to be of any help within the next 4 years. Okay, and then so in other words, the vaccines that hold the best chance of helping us with the COVID-19 pandemic are those that are currently in clinical trials. Okay, so now let's focus on the clinical trials. If you have a vaccine that's very promising, you're gonna apply to a health regulatory agency, and this usually takes a few months. This is not particularly time consuming. And what'll happen is you'll end up agreeing on a clinical trial design, and then you go and you execute that. All right, and then what happens then is there's 3 consecutive phases of human clinical trials. It typically starts with a phase 1 trial, or it has to start with a phase 1 trial. These are relatively small trials, and the emphasis is completely on safety, making sure that that vaccine appears safe. This takes about a year or more. So what I'll say, and what I have for these timelines, these are generally very conservative times that I have listed here, okay? So at least a year. Then typically it would move into a phase 2 human clinical trial if the vaccine appears safe. A phase 2 human clinical trial scales up. There's more people that are enrolled. And this again emphasizes safety, but there also has to be some evidence the vaccine is working. So perhaps maybe it has to demonstrate that it's inducing, promising looking immune responses. And this will typically take 2 years or more. And then the vaccine would migrate, if everything looks good, into a phase 3 human clinical trial. This typically lasts about 3 years. These are very large scale studies, and that will again emphasize safety, but also efficacy. The vaccine has to be demonstrated to work at this phase, or it will not be approved. So if a vaccine can transit through these 3 phases of clinical trial testing, then what happens is it can be approved for use, but then it has to be scaled up. And you see I've got a question mark here in terms of the timeline for this. The reason being is we have never in the history of mankind attempted to manufacture a vaccine on a scale that is needed for a global pandemic like the COVID-19. So we really don't have a timeline, but this is not trivial. This could take a very long time. People are arguing this alone could take up to a year. And then after the vaccine is manufactured, then there's the distribution administration phase. And the reality is we do not have an efficient distribution system set up, again, for worldwide delivery of a vaccine and getting this administered to people across the planet. So again, I've got question marks here. This is not, if you look at the testing that people are trying to do in countries, and the timeline it's taking to get testing done, that doesn't bode well for the distribution administration of a vaccine, which would rely heavily on very similar or even the same infrastructure that's being used for the testing. This could easily take well over a year. So in short, Traditionally, it has taken vaccines at least 10 years to traverse these clinical trial phases, so steps 4 through to 8. Okay, and what I want to point out is Merck, the big pharma company, Merck, to the best of my knowledge, currently holds the world record for the fastest time to be able to get a vaccine through this clinical trial research phase, and that was 4 years. And again, this is a record. This is an outlier. This is not typical of vaccine development. All right, and I believe they also probably hold probably the second fastest time to vaccine migrating through this system, which is 5 years for an Ebola vaccine. Okay, so there's just a certain amount of scientific rigor that has to be maintained. It's very difficult to try and go below this. Okay, and so what I want to point out then is, as we move on here, is some conclusions. All right, first, it takes a long time to develop an effective and safe vaccine. Now, efficiencies for COVID-19 vaccines can be built into the approval process, all right, through things like accelerated funding, streamlining the paperwork. There's an awful lot of administrative red tape. And also there's what regulatory agencies are doing right now is they're allowing some overlaps in the clinical trial phases rather than them running sequentially. The second phase is being allowed to start towards the end of the first phase, for example. But again, the efficiency should never compromise scientific rigor. And again, early vaccines that are in the early stage are not going to help us with COVID-19 within the next 4 years. And the COVID-19 vaccines that are in clinical trials likely won't have a substantial impact within the next 2 years, okay? And this assumes that they work well and are actually safe and receive regulatory approval. I hope I'm wrong, right? And this is something nobody can prove. Maybe we will have a vaccine, but I'm just trying to put forward what the realistic probability is, okay? And then some other things that I just want to go over with you quickly is some other things you have to think about when it comes to these vaccines, all right? Safety, of course, has to be paramount, both real and perceived. So I want to just point out a couple of interesting things, okay? So after the original SARS coronavirus outbreak, there were researchers who continued to conduct research on a SARS coronavirus vaccine. So that was the first virus. And what happened is in animal models, the vaccine looked very, very good. But what happened in those animals that were vaccinated and had very good looking immune responses, it turned out that the vaccine actually caused the severe acute respiratory syndrome to be more severe than in animals that had not been vaccinated. And this was because the vaccine had actually induced the wrong type of immune response, okay? The immune response that was induced actually promoted inflammation in the lungs. And so then when the animals were infected, they had this much more severe inflammation in the lungs than what the non-vaccinated animals would have experienced. And the other thing I want to point out is there's actually a historical precedent for a warp speed-like vaccine program, and actually that went quite awry, okay? So for those of you who don't know, I've got an article here where you can get more details if you want. But in the mid-1970s, there was actually an influenza outbreak at a military base in the United States. And what happened is several hundred soldiers became ill and one died. It turned out that was the only person who died. But it caused such a panic that there was this incredible race put on to develop a vaccine. This vaccine was pushed through the system very quickly. And the resulting vaccine actually only got distributed to less than 25% of Americans. But what's of concern is it caused 25 deaths and more than 500 people to become paralyzed, okay? So in other words, this vaccine was much worse than the disease. It caused a lot more damage than what the disease itself did, all right? And the other thing that I wanna point out is that even if a vaccine is truly safe, if the process to develop it gets interpreted as, quotes, cutting corners, that could negatively impact uptake, okay? And there's this disturbing number of polls that are starting to appear where they're showing that quite a few people in the ballpark of 20% of people who would normally be okay with taking a vaccine won't be willing to take a COVID-19 vaccine. And in some of these, up to 30% are undecided. So this could have a big impact on progress towards herd immunity, even if a very effective and very safe vaccine were to be made. Okay, and the other thing I want to point out is, unfortunately, a lot of governments, so the original goal behind this vaccine development program that's going on worldwide was to get a vaccine that would actually protect us from the disease. But what's happened is the bar has been lowered quite dramatically. such that a lot of governments and regulatory agencies will now define a vaccine as being successful and therefore worthy of licensing for use in people if it merely reduces the severity of COVID-19 and or prevents death. Right? So in other words, what we're talking about, if that's how a vaccine is going to function, it's really becoming a therapeutic intervention which would be used after the onset of COVID-19 and would not be a tool to eliminate it from a population, okay? And the other thing is we have to ask, are these COVID-19 vaccines going to work in the people who need it most? So elderly individuals and those who are immunocompromised or immunosuppressed are the people who are suffering by far the most severe cases of COVID-19 and the highest death rates due to COVID-19. And yet they're actually the people in which a vaccine would by far work the worst, okay? That's important to know. And the other thing is, so vaccinating young, healthy people that are around these people can help protect them in theory, right? But it's also important to know that even in the relatively young and healthy where these vaccines would probably work the best, the responses to vaccines follow a bell-shaped curve, and that means even among those who tend to respond best to the vaccine, there's always going to be individuals who are what we call low responders, and they do not respond well. So as you can see, all this adds up to a scenario where, well, an ideal, so I'll just state what I have here, right? An ideal COVID-19 vaccine that could accelerate progress towards herd immunity is one that would protect against the disease, provide the appropriate type of long-term immunity, that's the immunological memory. It would have a broad safety profile, including in those who need the vaccine the most, who are also the most fragile among our population. And it would also have to be broadly perceived to be safe, okay? Now, I want to highlight again, I nor anyone else can guarantee that a useful COVID-19 vaccine will not be available in short order, okay? It might be. And I certainly hope that I'm wrong in my predictions and that this does happen, but realistically, the chances of that happening are very slim, okay? It's got to get through the clinical trial pipeline, as I mentioned before, so we've manufactured in massive quantities and widely distributed in order to have a substantial impact, and I just don't see how this could happen within the next 2 years. And on that basis, I would advocate that a lot of caution be exercised when developing policies around the assumption that a vaccine will be available anytime soon. And so what I'm going to do is I'm going to stop there. But again, what I've done is for those who want to request a copy of this. Oh, sorry. Let me just say one more thing. Just to get for those of us who are doing this research, what I do think is realistic for the COVID-19 vaccines is developing technologies for what I would call the next big one. And what I mean by that is a so-called SARS coronavirus 3. So in other words, a dangerous coronavirus that may emerge in the future. And virologists will tell you it's not a question of if another dangerous coronavirus will emerge, but it's a question of when. And so what happens is we failed. As scientists, we failed to follow through with developing vaccines after the first SARS coronavirus. We failed to come up with a vaccine after the MERS coronavirus. And we can't afford to fail again after this one. If and when COVID-19 is in the rearview mirror, we need to stick to the program. We need to continue to develop effective vaccine technologies that can be vetted among the scientific community to identify those that are going to work the best, and ideally technologies that are plug-and-play compatible, so in the future we can quickly respond to a future SARS coronavirus. So again, I'll end there for the questions. But what I do want to do is, let's mention is, for policymakers, the media and investors, what I've done is I've actually put together here 2 slides. It's 9 questions, okay? And these are questions that a vaccine manufacturer should be able to answer if their vaccine has been well vetted. And I would certainly say before putting any trust or faith, or for example, investors putting your own money on the line with these, you should get these questions answered and to your satisfaction. If these questions cannot be answered to your satisfaction, I would really start questioning the quality of that vaccine. So are there any questions?
Yes, thank you, Byram. There are a lot.
Oh, okay.
I'm going to start with one myself for a moment, though, because over the weekend, because we're in lockdown here again in New Zealand, I don't know how closely you followed it, but Auckland is in a level 3 lockdown, which kind of means only essential work, but you're allowed to get takeaways and go to the supermarket. That New Zealand, the Minister of Health said that they were working on putting New Zealand near the front of the queue for the vaccine, or a vaccine, when it's ready. How realistic is that? And perhaps just to ask as well that if we continue to lock down, we're not going to be very well exposed anyway. So would that have any role in putting us closer to the front of the queue for a vaccine should one become available?
That's a great question. So you could argue it in a couple of ways. If the virus has not spread widely through your population, you could argue that New Zealand is in greater need than some countries who are closer to that herd immunity through a natural exposure to the infections. But typically what happens in these scenarios is that most people are going to focus on distributing vaccines, I would imagine, to countries that can document suffering the highest rates of severe disease and deaths. And so in essence, you may almost be shooting yourself in the foot by not trying to learn to live with COVID-19, right? Like other countries are, but by actually having these very strict lockdowns and very much limiting the cases, you're kind of limiting the data that you can show to make an argument that the vaccines are going to have a big impact on health and reducing severe cases and reducing deaths, right? I think that's going to maybe be the top priority. And ultimately, the reality is the only way for any country to ultimately guarantee that it's going to have rapid access to a vaccine that's developed is if they're developing it. And unfortunately, this is where we're seeing a lot of politics as well, right? So for example, I work in Canada. We have a case where an agreement actually was almost reached, got nullified because the other country that we were dealing with indicated the vaccine was being developed there, and they clearly indicated that their population would be the first to get it. And of course, Canadians would like to get access quicker, so they're looking for another partner. So there's a lot of politics involved. But yeah, by having a low number of severe cases and low death rate, you might actually be inadvertently putting yourself further down that line.
All right, I'll go back to the start. There's quite a few questions about the nature of immunity that are coming up in our list. There's been some dispute or lots of chat about whether it's antibodies or T cells that do the work, and early studies had found antibodies were not being produced, but then we discovered T cells are. I mean, what is the status there? What matters most, and perhaps why did we not know this to begin with?
Well, I'd say we do know what the most effective immune response is against viruses. Typically, there can be certain variations. We have to understand the viral biology, but in short, when you're dealing with viruses, you want both. You want both antibodies and you want T cells. The reason is antibodies are you definitely want antibodies for sure. Like if you had to choose between one or the other, you'd probably pick antibodies of the right type. The reason is what the antibodies do is if they're in the right location, they will bind to the virus and it'll do what we call neutralize the virus. So it'll prevent the virus from ever infecting a cell. So when the SARS coronavirus comes into our lungs, if we have neutralizing antibodies there, they'll bind to the virus and prevent it from infecting the cells in our lungs. If the virus can't get into the cells in the lungs, it can't replicate. And so we get rid of the viral infection. These virus and antibody complexes will be cleared and we clear the infection. But the reason why you want T cell responses is if any viruses get past that antibody barrier, if you only have antibodies and you get any viruses that get past that barrier, once the viruses want to live inside cells, they have to be inside cells in order to replicate. So, and antibodies cannot get inside cells. So if you have any viruses that get past that antibody barrier and into the cells, you have to have T cells in order to get rid of those virus infected cells. That's why you want to have both. And what's actually been frustrating me a lot as a scientist, I've been very surprised because, so first of all, I'm not particularly surprised when companies release information. The reality is the information is publicly released by companies. the scientific information is really lacking. The details are so lacking that scientists can't properly assess the quality of these vaccines. And then what happens is if you, and then a lot of people have been focused on antibodies and reporting this, but even the scientific literature, as these things are being published, I'm not seeing the scientific data that I would like to see, which is what are the types of these responses? So we're hearing that there's antibodies being induced and or there's T cells being induced. But for example, when it comes to antibodies, like I mentioned, there's two categories of antibodies. There's antibodies that are ideal for targeting extracellular pathogens like bacteria. And the reality is if you have an extracellular pathogen, those are the types of pathogens that actually induce the highest magnitude antibody responses. So we get very excited because we see a high magnitude response. The reality is that a natural antibody response against a virus tends to be of lower magnitude, but they're the right types of antibodies. They're the ones that can neutralize viruses. Worst case scenario, and this is actually, so that study that I mentioned where a SARS coronavirus, that's the first SARS coronavirus, when the vaccine was ultimately tested in animals, it actually made the disease worse. People thought, the scientists thought it was going to be great because they had great antibody responses, but these antibodies were designed to target extracellular pathogens. It's because the vaccine was being perceived by the immune system as it was thinking the virus was an extracellular pathogen because the virus was inactivated, so it couldn't actually infect cells and mimic the natural infection. So the immune system responded like it was an extracellular pathogen, produced great antibodies, but those antibodies, what they actually did was they actually promoted inflammation because they were wrong type of antibodies. So I just want to emphasize, we want antibodies and we want T cells, but we need the right types of antibodies. And the problem is if you just look in the blood doesn't particularly help because the virus isn't infecting through the blood. We have to remember that. We need the right types of antibodies in the lungs, right, where the virus is actually infecting the body.
Right. Now, if I get you right, you're saying that the work done on the SARS-CoV virus and earlier ones, vaccine work, that it's given no advantage to the current studies?
Well, it has given advantage. So that's why there's some vaccines that could quickly be put into clinical trial in many cases. But one of the concerns is what happened in large part. That's why I just want to emphasize towards the end of my talk that we have to, quote, stick with the program. What's going to happen is if COVID-19 ends up in the rearview mirror, it's no longer a huge problem. Our economies are going to be wrecked, right? And when economies are wrecked, often one of the places where dollars are diverted from is research, because research tends to be a luxury, right? But we have to continue with this because that's exactly what's happened with the previous coronavirus outbreaks. Is first of all, if you can imagine, if you're a large pharmaceutical company that's invested a lot into a vaccine and that outbreak is now over, you're not going to have any vaccines to sell. So they often then focus on what the next problem is. And those who fund, for example, academic research programs are also often focused on what are the current problems. And so when a problem is in the past, it's hard often to get funding for it. But government agencies, everybody who's working on this vaccine has to continue. We can't have that attitude again that we'll worry about it if it happens in the future. We have to understand, we now have a history that suggests that we're going to get a dangerous coronavirus maybe every decade, approximately once every decade. And so we have to get these vaccine technologies fully vetted and not drop them after the pandemic has ended. And there's so many vaccines that are being developed. We have a golden opportunity to spend the next several years testing all these head to head and finding out which are the ones that actually look really good. Because the reality is whatever comes through the clinical trial pipeline right now almost certainly is not going to be the best COVID-19 vaccine. And in fact, we have to be a little bit careful because, as I mentioned, if we're dropping the bar too low and saying we don't even need a vaccine that's going to be protective, if we can just find one that's going to help reduce the severity of the disease after somebody's infected, the problem with that is that's going to occupy the manufacturing and the distribution pipelines and divert resources away from the better vaccines that are sitting in the background. So we have to make sure that we continue to work with the vaccines that aren't as advanced.
So it sounds like what you're saying is, given that time frame, because I think you said that no vaccine within the natural curve, or not demise, but the reduction of the epidemic. No vaccine within that timeline. Is it possible then that the money runs out, or the commercial advantage of producing one runs out because the demand goes away, and that's the companies will just stop? Do you think this time?
Well, it's interesting. It's interesting to say that, because of course, as these vaccines are starting to move through the clinical trials, again, there have to be more and more people recruited. And once it gets to a phase 3 clinical trial, there's no question what has to happen in a phase 3 clinical trial is volunteers who are vaccinated. We have to be able to demonstrate that that population of vaccinated individuals experience lower rates of COVID-19 than those that are not vaccinated. And in order to do that, you need a pretty robust exposure, among a population in order to be able to run that study. The lower the case rate, that's just going to make the number of people that are required to properly test the vaccine balloon. And so we're actually running into a problem in a lot of countries where there simply aren't enough cases. So in Canada right now, we have all kinds of funded COVID-19 research projects, and we're actually struggling to get people with COVID-19 recruited to do them, right? And it's going to be the same thing with a vaccine, because there just isn't enough of it circulating in the population to ensure that vaccinated volunteers get properly exposed to it, to see if it protects.
Right, and after that, you run into ethical problems about introducing the virus.
Exactly. Yes.
Okay.
Which honestly, I wouldn't want to be the person to do that for what I just said. We have to be careful. There's this antibody-dependent enhancement of disease, right? There are precedents for if you haven't designed the vaccine properly, right? It might look safe in a volunteer. Remember, with that vaccine I told you about with the original SARS coronavirus, there were no safety issues at all in vaccinated animals. It was safe in the vaccinated animals. It made the disease worse once they got exposed to the virus, once they were challenged with it, right? So we have to be very careful. So I wouldn't want to, I don't think it's a good idea to intentionally try and expose people to the virus for that reason.
Okay. So therefore, we can't skip any of the important parts of the vaccination development, particularly the trials. We just can't do it. We don't want to hurry.
Mm-hmm.
Okay.
Yeah. Like I said, there are efficiencies that can be built in, but only so much, right? So if 4 years is the record, to me, it's like, for a sprinter running the 100-meter dash, right, the world record is in the ballpark of 10 seconds, right? To expect them to cut that down to 5 seconds, like, there's just physical limitations. And like there's physical limitations in the human body, there's physical limitations in that vaccine timeline that I showed you. This pipeline that things go through. You can only shorten it so much. You can only find so many efficiencies, right? In order to maintain scientific rigor, there's a minimal amount of science that has to be done. And I just can't see that being completed in a way that could see a vaccine widely distributed within the next 2 years.
So does the same go for the types of vaccines you were talking about, which aren't really a vaccine? They just lower they lower the condition, the experience of the condition.
Well, I think it's kind of the desperation. You see, so what I'm worried about now is there's been, like we know, there's been billions of dollars invested in the development of vaccine. The reality, let's face it, if we're just very blunt about this, if a vaccine does not become available to help us with COVID-19, and we do find that countries just naturally, that they work hard to learn to live with this virus in a relatively safe fashion. So like the previous speakers were talking about, so yes, we experience cases, but they don't translate into very many severe cases. They don't translate into very many deaths, and we are able to achieve herd immunity that way and that naturally. And a vaccine has never been developed in time to help us with that process. The reality is, right, we're going to look back on this, and it will have been millions and millions and millions of dollars wasted, ultimately, in many ways, except for the continuation of trying to develop those for a future vaccine. But like I said, with that said, most of that money is the organizations that have the greatest ability to push these vaccines through the clinical trial pipeline, who have the infrastructure in place, are usually the big pharma companies. And like I said, it just doesn't make any business sense to continue developing a COVID-19 vaccine if COVID-19 is no longer a problem, right? So we're kind of in a situation where we almost need COVID-19 to last long enough for there to be vaccines, or there's going to be a lot of investors who are going to be very unhappy and a lot of people running businesses who are going to be very unhappy, right?
So is a vaccine actually possible? You know, given the previous work that you would say didn't result properly and what was aimed for, is it if you worked long enough, would you get one? So either this coronavirus or a following one.
Okay, so it's interesting. I think this goes back a little bit what Dr. David Katz was saying as well, right? I would say, going beyond that, even if there is a good and safe vaccine that is made, again, there's no precedent for getting it manufactured in sufficient quantities and distributed worldwide in time to have a really big impact, and worse. So I just mentioned that in terms of, like, a lot of organizations now are redefining a successful vaccine as being one that basically gets used as a therapeutic agent after infection, right? It's not going to prevent infection. But then Dr. David Katz mentioned, and this is absolutely true, for those vaccines that are still aiming for preventing infection, the definition of a really good one would be one that's 50% effective, right? At this point. And as you mentioned, so I mentioned, getting back to the herd immunity, if people are saying, are correct in that we need at least 70%, if not 85% of a population to be immune to achieve that herd immunity and stop the spread of the virus, then, you just start putting so if the vaccine was 50% effective, you're only getting 50%, right? That's the whole point. And that's why he was saying that, why that's an issue. That's not getting you to herd immunity, right? And then if you add on to that the fact that there's a growing number, because of these crazy terms that have been applied to this, right? Using warp speed in a vaccine development program does not confer a lot of confidence with a lot of people. And in fact, it's caused a lot of harm, right? Like I said, you look, you do the searches, there's all kinds of surveys being done now. And on average, they're showing about 20% of people who would normally take a vaccine won't take the COVID-19 vaccine, just because they're thinking that the process sounds like it's not the safest. Yeah. So if you have a vaccine that would only be 50% effective in a population, and 20% of the population isn't even going to take it, and then you have the relatively small number of anti-vaxxers that would be on top of that, and of course, you're just not going to achieve herd immunity anyways. So I think this has to be recognized. So I just really think that waiting for a vaccine, even if this does come through quickly, I still think when you add up all of the facts, it's not the right thing to lean on when making policies. So
in New Zealand situation, we've got a closed border. This is the final question, Byram. We've got a closed border. We've locked down because we found some new cases. Should we be waiting for a vaccine, or should we be trying something else?
So again, the hard thing that I will admit is no vaccinologist can say with 100% certainty that there won't be a vaccine. But like I said, if you look back at my presentation, you look at all the considerations, all the people who won't respond well to a vaccine, the people who won't be willing to take a vaccine, the unknown length of time to distribute it and manufacture it, outside of the long time it typically takes to migrate through the clinical trial pipeline, it all adds up to the fact that you got to do cost-benefit analysis. So it's the chances of one being available the next 2 years is extremely slim. I can't say zero, but extremely slim. And I don't think most people would want to develop policies for their entire country on something, on a chance that's extremely slim.
All right. Thank you, Byram. Thank you. Look, we wish you good luck with your own efforts. This isn't the first coronavirus, and it won't be the last. So we need people like you doing the work over the long term. So good luck with that. Thank you for coming on. Actually, Byram, if you're able to stay for just a little bit longer, there are a lot of questions on the Q&A list. Some of them would deserve a written answer quickly, if you're able to. If not, we do have some other experts on the panel who can probably have a go.
Okay.
Thank you
very much for having me. Appreciate it.
Byram.
Take care.